guys, today I'm going to talk to you about regulation of carbohydrate metabolism. I made this Word document as kind of just like a little flow through it. We're going to talk about the regulation of all the different cycles involved in carbohydrate metabolism. So that's going to include glycolysis, gluconeogenesis, glycogenolysis, and glycogenesis. So we're going to get started and we're going to talk about glycolysis first. So phosphofructokinase 1 is the rate-limiting enzyme of glycolysis, and it's going to be very highly regulated. This is the reaction that phosphorylates fructose 6-phosphate to fructose 1,6-bisphosphate, which ends up making the molecule symmetrical to be cleaved. And if you remember, the delta G of it is very negative. So it's going to be highly regulated because it's considered one of those committed steps of glycolysis. Its inhibitors are going to be ATP and citrate. If we think about this logically, this makes sense because the point of glycolysis is to break down glucose for energy. So if we have a high amount of ATP in the cell, then we don't need that energy, so we're not going to break down our glucose because that's the whole point, is to produce energy. Citrate is another one of those molecules. We haven't talked about the citric acid cycle yet, but it's a molecule that's involved in the citric acid cycle, which is also used to produce energy. So if you have a lot of citrate, you can use that in the citric acid cycle, and you won't need to break down pyruvate to then turn it into acetyl-CoA, which then gets turned into citrate because you already have that citrate available. So in the opposite vein, what's going to activate your phosphofructokinase 1 is going to be your ADP or AMP. So this is going to signal that you have low ATP and not enough energy for your cell. Another molecule that's important is fructose 2,6-bisphosphate. This is solely a regulation molecule. It's not broken down or created in any sort of a cycle. It just flip-flops, and it is considered just a regulatory molecule. So there's a nice little flow chart down here from your lecture notes, which shows just a visual drawing of what is going to activate and inhibit this specific reaction, turning your fructose 6-phosphate into your fructose 1,6-bisphosphate. So now we're going to talk about some hormones that regulate metabolism. So when you have low glucose levels in your blood, your body is going to release a hormone called glucagon. It's produced in your pancreas, in the alpha cells in your islets of Langerhans. This is going to inhibit glycogenesis and glycolysis and stimulate gluconeogenesis. So the reason behind this is if you have low blood glucose levels, you're not going to want to create glycogen in your cell or break down glucose. You're going to want to make glucose so that you can release it into your bloodstream to increase your blood glucose level. Okay, so if you are at a state where you need some energy, let's say you haven't eaten anything and you're running to class to try and make it on time, your body's going to produce epinephrine. So this is produced in the adrenal medulla, and it causes the release of glucose by the liver. It inhibits glycogenesis and it stimulates glycogen breakdown. So this is going to be when you don't have any when you're not intaking any glucose, you need to take it from your glycogen stores. So you're going to not be making any more glycogen, you're going to inhibit glycogenesis, and you're going to stimulate glycogen breakdown so that you can get some glucose in that can be used for energy by your cells. So your liver is going to release that. Like we said, the liver is your main store of glucose, and it's going to release it into your bloodstream to send to your other tissues that need that glucose to break it down for energy. So in the opposite vein, when you have high blood glucose levels, your body's going to produce insulin. So this is also produced in the pancreas, just like glucagon is here, but it's produced actually in the beta cells in the islets of Langerhans. This is going to be a fact that you're going to want to be familiar with because that's a very simple question to ask: what cells in the pancreas produce insulin versus what cells produce glucagon? And it's just the alpha cells for glucagon and the beta cells for insulin. So the release of insulin, like we said, if we remember, that's a hormone that acts on the receptor tyrosine kinase cascade. So this is going to cause uptake of glucose from the blood because you have too much in your blood, so you're trying to get rid of that high blood glucose level. And this is going to inhibit glycogenolysis, and it's going to stimulate glycogenesis. So you're not going to be wanting breaking down glycogen. You're going to want to be increasing your stores. So it's going to stimulate glycogenesis. All right, so we talked a little bit about hormonal control, and now we're going to go back to another regulator of glycolysis. So this is going to be pyruvate kinase. So most of the regulation steps we're going to see are involved in the molecules in which it's considered a one-way path. So if you remember, pyruvate kinase catalyzes the reaction in glycolysis, which transfers the phosphoryl group from that phosphoenolpyruvate, that really high energy molecule, to ADP to form ATP. So this is a very negative reaction because you are transferring that phosphoryl group and you're moving from a high energy state of phosphoenolpyruvate to a low energy state of the pyruvate that you create. So this is going to be one of those steps that's going to be highly regulated. Its inhibitors are going to be ATP because, again, if you have a lot of energy in your cell, you're not going to be wanting to break down glucose because you don't really need it, because the sole job of glycolysis is to produce your pyruvate, which can then enter your citric acid cycle, which is going to produce you some ATP. So if you already have it, you don't need to go through glycolysis. Another inhibitor is going to be acetyl-CoA, and this is because you use that pyruvate to make acetyl-CoA. So if you already have a high amount of acetyl-CoA, you're not going to need to convert that pyruvate to it, so you won't need to make pyruvate in the first place. And this is what we'll see in a lot of these pathways, is that the molecules a little bit down the road that are produced, a little bit downstream in the deeper processes, will sometimes inhibit the production of the earlier molecules, because those earlier molecules are what are used to produce them. So if you already have those, you don't need to produce the earlier ones. So another one that inhibits pyruvate kinase is fatty acids. What we're going to see, we haven't talked about it yet. This won't be till the next test. We're going to talk about fatty acid beta oxidation, which is the breakdown of fatty acids for energy. And what we're going to see with that is that fatty acids produce an extremely, extremely high amount of ATP per every molecule broken down. I know that glycolysis, when all is said and done, and you go through the citric acid cycle and electron transport chain and oxidative phosphorylation, you have about, I think, 34 to 38 ATP produced, and the fatty acid number, I believe, is somewhere within the at least in the higher hundreds, I believe, is the number. So you get a lot more energy from breaking down fatty acids. So if you have a high concentration of fatty acids, your body is going to prefer to use that for its energy, because it does increase and it does release so much more energy than breaking down a molecule in glycolysis. Okay. So the activator for pyruvate kinase is going to be fructose 1,6-bisphosphate. This is that molecule that we produce that is from the phosphofructokinase 1 enzyme reaction. This is that symmetrical molecule that we end up cleaving into the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And this makes sense that this would activate pyruvate kinase, because this is one of those committed steps. So once you make this fructose 1,6-bisphosphate, you're committed to going through glycolysis and breaking it all the way down to pyruvate. So it makes sense that an increase in abundance of this molecule would activate an enzyme further down in the chain. And again, this is something that we're going to see a lot in these processes. So next we have some molecules that are in charge of regulating both glycolysis and gluconeogenesis. So we have two enzymes. We have PFK1, phosphofructokinase 1, which we've already learned about, and then we have FBPase1. So phosphofructokinase 1 we talked about before, but we have not talked about FBPase1 yet. So FBPase1 is an enzyme that is going to be responsible for it's going to be responsible for that second step of gluconeogenesis. So it's going to be responsible for dephosphorylating that fructose 1,6-bisphosphate to fructose 6-phosphate. So the opposite direction, the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate, is by PFK. So it makes sense that these two molecules are going to kind of be in opposition to each other, okay? So when PFK1 is more active, FB-PACE1 is going to be less active because glycolysis and gluconeogenesis in these two reactions, this one which is in glycolysis and this one which is in gluconeogenesis, they directly oppose each other. This enzyme here, FB-PACE1, we learned is the bypass step for this reaction because it won't go in the opposite direction. So we talked about the inhibitors and the activators of PFK1 before, but we're going to talk about the inhibitors of FB-PACE1. So FB-PACE1 is inhibited by AMP because FB-PACE1 is usually used when you already have enough ATP in the cell, and so you're creating glucose for some reason. So just like ATP is going to inhibit PFK1, AMP is going to inhibit FB-PACE1 because you'll probably already have enough glucose since you're signaling you have a low amount of energy in the cell. So we saw that fructose 2,6-bisphosphate is an activator for PFK1, and in the opposite vein, which makes sense, it's going to be an inhibitor for FB-PACE1. So if you have an increase in F2,6BP, your FB-PACE1 is not going to be very active, and you're not going to be producing glucose and gluconeogenesis. And in the opposite vein, your PFK1 is going to be active, and you'll most likely be doing a lot of glycolysis and breaking down glucose. So, like I said before, a major part of the regulation of glucose and gluconeogenesis is this control of that fructose 2,6-bisphosphate, which is a regulation molecule. You can control it in two ways. You can control it with a bifunctional enzyme. So, fructose 2,6-bisphosphate solely serves as a regulatory molecule. When you have low fructose 2,6-bisphosphate levels, you're going to have a decrease in glycolysis and an increase in gluconeogenesis. And when you have high fructose 2,6-bisphosphate levels, you're going to have an increase in glycolysis and a decrease in gluconeogenesis. And unlike we've seen before, in the other regulatory molecules, these are mostly what are going to be involved in those cellular processes, right? So citrate is involved in glycolysis, and you have ATP and ADP through all those reactions being involved. But fructose 2,6-bisphosphate is solely a regulatory molecule, so it's just going to basically be controlling which cycle is active, but it won't be used within the cycle. So that's something important to remember. So this bifunctional enzyme that we can control the fructose 2,6-bisphosphate levels with is going to be a molecule that has two functional units, and each unit performs a different activity, and the activities are actually opposite of each other. So the molecule that we're going to see, if we see right here, is composed of this PFK2 subunit and this FB-PACE2 subunit. And the PFK2 subunit is going to be responsible for phosphorylating things. Remember, it's protein kinase, or it's a protein kinase, phosphofructokinase 2. So kinases are responsible for phosphorylating things, and then the other part of the molecule, the FB-PACE2, is a protein phosphatase, which is responsible for dephosphorylating things. So this is going to be fructose 1,6-bisphosphatase 2. Okay, so this the way it controls phosphorylation, so the enzyme itself, I'm sorry, I misspoke. The enzyme itself is controlled by phosphorylation. So the enzyme itself is going to be phosphorylated by a protein kinase or dephosphorylated by a protein phosphatase. Okay, so when the enzyme is phosphorylated, your FB-PACE2 is going to be active and your PFK2 is going to be inactive. Okay, so here we have the phosphorylated form that's been phosphorylated by a protein kinase, which is going to happen as a result of glucagon. So you're going to phosphorylate this, the hydroxyl group of this enzyme, and you're going to have FB-PACE2 active and PFK2 inactive. So if you have this in this molecule in this situation, you're going to have a decrease in fructose 2,6-bisphosphate because this FB-PACE2 is going to dephosphorylate it. So you're going to inhibit glycolysis and you're going to stimulate gluconeogenesis, which makes sense because if we look here, we have this glucagon, which is going to be the main activator for this protein kinase. So when we have glucagon activating this protein kinase, we're signaling to our body that we do not have glucose and we need it. So we're going to be making glucose through gluconeogenesis. In the opposite vein, when we dephosphorylate this enzyme with a protein phosphatase, the PFK2 form is going to be active and the FBPase2 is going to be inactive. So this is going to stimulate glycolysis and inhibit gluconeogenesis by increasing the fructose 2,6-bisphosphate levels, okay? And this makes sense because when we have a high amount of insulin, we're signaling to our body that we have more glucose than we need, and we need to break it down through glycolysis, and we're not going to want to be making more glucose because we already have enough that we need to be breaking it down. Okay, so this is where the hormonal control of the fructose 2,6-bisphosphate comes in, and this is just nice to see the flow of it. A lot of the questions that you're going to be asked on the test are going to be asking you about the flow of what gets phosphorylated and what activates what, and go through the chain of each process. So this is a good way to connect it all. So what's going to happen is when you have low blood glucose, your body's going to release glucagon. Your protein kinase A, which we saw was activated from that GPCR, the G protein coupled receptor cascade, which is activated by glucagon and epinephrine. So glucagon is going to activate that PKA, and that's going to be in charge of phosphorylating that bifunctional enzyme. So when you phosphorylate that bifunctional enzyme, you're going to activate the FBPase2 subunit of it and inactivate the PFK2 subunit. So it's going to be dephosphorylating this fructose 2,6-bisphosphate and lowering its levels, which is going to, like we said, inhibit glycolysis and stimulate gluconeogenesis. Again, this makes sense because glucagon, you're signaling low blood glucose, so you want to stimulate the creation of glucose and tamper down on the breaking down of it because you already don't have enough. So with insulin, when you have high blood glucose, you're going to have that receptor tyrosine kinase cascade, which ends up having this protein phosphatase 1 enzyme, which phosphatases, we remember, dephosphorylate. So this is going to be the enzyme that's in charge of dephosphorylating your bifunctional enzyme. So when your bifunctional enzyme is dephosphorylated, you're going to activate the PFK2 subunit and inhibit the FBPase2 subunit. This is going to in turn increase the fructose 2,6-bisphosphate levels, which is going to stimulate glycolysis and inhibit gluconeogenesis. This makes sense again because insulin is telling you that you have high blood glucose, so you're not going to want to make more with gluconeogenesis, but you're going to want to stimulate glycolysis to break that down. So I'm going to be posting this document just on the Files tab without this voice recording so that you can look it over, but this one I'm going to put on the Pages tab. So after you listen to this, make sure that you go back through your PowerPoint notes and make sure that you really understand the regulation, and feel free to ask me any questions if you'd like. I'm going to be making a PowerPoint for the citric acid cycle within the next couple days, which I'm also going to narrate, so hopefully this works out well and it helps you guys. Like I said, email me with any questions you have. I will be happy to answer them. I hope this helped you, and good luck studying.